Okay, so now let's turn our attention to biological water problems. Here are our three categories: pathogens, okay, such as Pythium, Phytophthora, and really we don't have a threshold of how much is a problem, although the phytopathologists are working on exactly that issue right now in a number of different universities. The problem is it can lead to plant disease, especially if we've got a stressed plant, we're over-irrigating, so we've got the whole disease triangle going for us, or against us. Bacterial levels. Well, a guideline is if you have in every 1 milliliter, which is a cubic centimeter of water, more than 10,000 colony-forming units, okay, so 10,000 bacteria that are going to form colonies, then we tend to see problems of clogging of emitters, and it's an indicator of high biological load. So you can also use bacteria counts before and after a water treatment to see whether that water treatment is working. Biofilm and algae levels, there's no clear threshold available, but we can see when issues are occurring. And again, it can contribute to clogging of emitters, can be a worker hazard, can disrupt irrigation and wettability of your growing media, and can lead to pest levels such as shore flies, for example. And the treatments in all of these cases for these biological issues are filtration to reduce organic load, and followed by a sanitizer, and there are a number of those available that will be talked about later in this webinar series. So here is a healthy root system and a diseased root system. This photo from Dr. Rob Wick at University of Massachusetts. And in these liners, if we just take a small sample here, which Dr. Wick took, and put under the microscope, then these are sporangia, and inside every one of those sporangia, they can release a number of zoospores, which are the swimming infective spores of Pythium that can cause root diseases. So what do you do to sample your water for plant pathogens? There's a couple of options. One is you can send your sample off to a laboratory such as University of Massachusetts. They will bait the water. So these are eucalyptus leaf discs that have been put in your water sample, and the zoospores swim towards those leaf baits, and then they put them onto agar and plate them out and see whether they're going to grow fungal colonies on this particular agar, which is going to be appropriate for the growth of Pythium or Phytophthora. Now that is only an identification to the genus level, and not all of these species are going to be either pathogenic to plants or pathogenic to the specific crops that you're growing. So you may want to get a sample sent for a more detailed analysis. For example, University of Guelph provides a DNA scan service. You send your water sample off, and they will check whether the DNA for about 70 or more known pathogens are present in your irrigation water. One of the key differences is that the plating method will tell you whether these are live organisms. This will just tell you whether they are present. So if you've got a water treatment system, you may have the DNA present, but the organism may be dead. Last week we had a presentation from Jennifer Park, who's a plant pathologist at Oregon State University. She's done a lot of work with testing for waterborne pathogens with ELISA kits, where you get a water sample, you have leaf baits in them, just like in the previous slide. You then grind up the leaf baits in an extractant solution and put a droplet onto what looks like a little pregnancy test for some instant results. And her experience has been that there's a lot of cross positives between Pythium and Phytophthora in the ELISA kits that are currently available. So this is really kind of under development as a grower tool. Another biological problem is biofilm. Okay, and you can see this lovely mister here, which is obviously not going to be doing a very good job of spraying out onto the crop, and it's also going to cause dripping down on the crops below. I'm sure you don't have anything like that in your greenhouse. You can monitor biofilm directly just by looking at those emitters, counting clogged nozzles. You can also put a window into your irrigation line. You can see whether there is biofilm, or in a line that has fertilizer, it's probably going to be a complex of algae and biofilm. And Dr. Peter Conjoyan has done a lot of research work on understanding biofilm and its control, especially with chlorine dioxide. So you can put ports into your irrigation lines to see how much biofilm is actually growing. You can also take a water sample and send it to a lab, and they will plate out and do these colony counts of colony-forming units per milliliter, typically of bacteria, which make up a lot of the biofilm. Now, one of my students, Dustin Metter, has developed a very nice technique where he takes a sample of water, puts it onto a Petri film, which has got a dehydrated nutrient solution on here, and then puts it at room temperature into a dark environment, such as a drawer in an office, and waits for 3 to 5 days, and then counts the amount of bacteria or yeast and molds, because there's a couple of different kinds of Petri films available. And Dustin's provided some training to a number of growers to use this technique, and it's really quite easy to use. And the basic idea there is the threshold of 10,000 colony-forming units per milliliter of bacteria indicates a high biofilm risk. So here are some data that Dustin took at different points in an irrigation system. And this would be the kind of sampling that you could do to look at bacterial loads as well as presence or absence of pathogens through your irrigation system. This is the source closest to the municipal or well supply. This is the furthest outlet away in the nursery, away from that incoming water. This is a concrete-lined tank indoors. These are ebb and flow benches or concrete floors. And this is an uncovered catchment basin outside. And you can see that the difference from the source to the first outlet represents the biofilm that's growing in the lines. And anywhere you're storing water, you've got very high microbial load is likely to develop, especially if there's a nutrient source. And it's likely that if you use those irrigation sources without some kind of treatment, at least with filtration and probably a sanitizing agent, if you're using drip irrigation or misters, you're likely to get clogged emitters. Now, emitters can clog for other reasons as well. We've talked about the biological sources of clogging. Another thing you can do is you can take those emitters and put them into an acid solution. We're going to suggest vinegar just because it's easy to handle. And if it's a chemical such as calcium magnesium deposits, then you leave that overnight, you're likely to see that much of that chemical is going to dissolve and clean up the emitters. It could also be particulate. So it could be that you see little bits of sand and silt that fall out. So here what you may find is, okay, we've got a chemical problem here, as is the case in this example with this is iron. And so we need to probably drop pH in our irrigation water or remove the iron before it gets into the irrigation lines. If it's particles, it's basically going to be a filtration issue. Now, algae can be a problem on growing media surfaces, on floors, and so on. It can also be certainly a problem in ponds and concrete floors. Anywhere we're recirculating water, we're likely to run into issues. Now, you can send algae off to a specialist lab. So, for example, we recently sent the sample from a sub-irrigation tank from a Florida grower off to Green Water Laboratories. Had a great service. I think it was over $150 for the sample. But they came back with an indication of the algal types and the quantity of the different types of algae. Okay, because there's a complex of many organisms that could be made up in your sample. And knowing that algae makeup could be useful, perhaps, in selecting algicides. I don't think most growers are going to go to the point of sending samples into an algae testing lab, but it could be done. Now, there is a very nice extension bulletin. If you Google Purdue extension and the authors, such as Roberto Lopez, Diane Cambarato, and look under controlling algae in irrigation ponds, then it goes through, for example, how different algae types tend to respond to different algicides. What we do in the greenhouse in our research work, this is some an example of a trial that Javier Lopez on my team is running. And here he's got some poinsettias and some coleus and torrenia with different mist solutions going down onto crops. What he does is he takes a little plug of growing media after we've run these different solutions over time. We do an extraction, and Gavin Mohammed Poor and my team tends to do that, the lab side of things. They extract the green solution here with a solvent, and then they put it into a spectrophotometer and measure how green that solution is. And that's an indication of how much chlorophyll and how much algae that there are. There is a kit available for on-site use, about $200 at lamont.com, and it'll do kind of up to this point. You extract a solution, you filter it, and you can look at how green it is. But I'm not sure really how useful that is. Maybe it is a relative measurement. Here's an example of an experiment where we had, through a mist system, either clear water with no fertilizer, fertilizer by itself, fertilizer with copper ionization 2 parts per million, or fertilizer with copper sulfate at 2 parts per million. Okay, and the fertilizer was 17,417 at 75 parts per million nitrogen in the mist for 2 weeks. And here's the chlorophyll levels that you can see from those soil extractions. So we can see that the worst case scenario was fertilizer only. The best was no fertilizer in the mist. And if we added copper ionization or copper sulfate, then we got some control of algae. Now, one of the problems is that we consistently see, not just with copper, is when we have enough of a sanitizer to really have a strong impact on controlling algae, then we're likely to get some phytotoxicity in certain sensitive plants. Here we've got some osteospermum. And so basically the key to algae control, partly, yes, we can use a technology such as copper ionization, but it also comes to water management. So if you want to really evaluate your algae control system, look at how much water is leaching out from the bottom of your propagation trays or pots, collected with a collection tray for a day or longer. And the more water that's washing through those pots or trays, the more it's going to indicate overwatering. Okay, so algae control comes down largely to water use.